what I'm going to talk about today is what role do animal models play in developing the next generation melanoma therapies. It was so inspiring to hear the talk before, and I really appreciated when he highlighted that drought region. But what was actually happening during that drought region is we're trying to do research, and understand new concepts and ideas. And so what I'll talk about today is how, as a community, we use animal models to try to think of the next ideas for therapy in melanoma. And I'll focus a little bit on some of the work that we do. We primarily use this model system, the zebrafish, and I'll tell you a little bit about why that is. So many of you, of course, will know that melanoma is a cancer, but where that melanoma comes from is from our melanocytes. And those are our pigment cells, and these color our hair, skin, and eyes. And I'll come back to this theme about how important it is that melanoma comes from a melanocyte at a few points in my talk. But I just wanted to talk about what healthy melanocytes do. And what they do is they protect us from the harmful effects of UV irradiation. And so people with lighter skin are found in regions of the world where there's less sunshine, and people with darker skin are found in regions of the world where there's a lot of sunshine. And that's because what the melanin is doing is protecting us from the UV, but the other issue is that in the northern and very southern hemispheres, we need sunshine for vitamin D, and our skin makes, that's where we get the UV to generate vitamin D. And so through evolution, as human populations moved away from the equator, they lost some of this melanin so that they could get enough vitamin D. So there's this balance that melanocytes are trying to do to protect us from the sun, but also make sure we get enough vitamin D. So with that little introduction about where melanomas come from, these cells, I just want to come back to this question about why are model systems important for melanoma research. And there tend to be two broad camps about who's doing research. So one side is this basic or foundational science, sometimes called discovery science. This can often be blue sky thinking. This is sometimes science where we have no idea where it's going to go or what the outcomes will be. So this is what we consider basic science. And then there's also clinical science. And some of you may be engaged in this clinical science, clinical trials, thinking about samples, trying to understand the disease in the person and through the trajectory of their treatment. But there's a very important component that links these two. And this is what's called translational science. And I have to say, this is what MRA is especially good at funding, which is trying to link these two. So these are innovation, new ideas, sort of really novel concepts, don't quite know where it's going yet. And here we know exactly where it's going. And translational science really allows basic scientists to think about how they can direct their work toward important clinical questions. But then it's also very important for going the other way. There's important observations seen in the clinic. How can we then design experiments to try to understand what's happening? And this is where model systems really come in. And in particular, animal model systems, they allow us to ask questions that may be relevant in the clinical setting. So what are model systems? Well, model systems are broadly living organisms that we can use in the lab and that are tractable, that we can do experiments on relatively quickly. So some of these include yeast cells, flies, worms, plants, but there's also animal models, which are zebrafish, which I work on, for example, mouse and frog are examples. And the reason why we can use model systems and make progress in science is because the fundamental biological processes are shared in cellular life. And in fact, some of our most profound insights into how cells divide and misregulated cell division is at the heart of all cancers come from yeast. So we really, because we have this shared DNA and this shared biological processes, it means that what we find in some of these very basic organisms can be relevant. Model systems allow us to do discovery. So they allow us to test new ideas very easily. They allow us to test function. And I'll give you an example of how we did that when I first started studying melanoma. And they allow us to test hypotheses. And so if we have new hypotheses come from the clinic, we can test those in model systems. Importantly, in particular with vertebrates, which include fish, we share a vertebrate body plan, and with mammals, our disease genes are shared. So even in a little zebrafish, actually 70% of our DNA is shared with a zebrafish, and 80% of disease genes are shared. So even though we look quite different, actually in evolutionary terms, we're actually quite similar. Ease of manipulation. This is why we have model systems. This means that I can go into the lab, I can inject them with genes, I can inject them with new molecules, I can study them under the microscope, I can add drugs to them, and I can see what happens. And really importantly is we can use large numbers of model systems, and they are relatively clonal. So humans are very different from each other, but when we use a model system, what we try to do is keep them as similar as possible so that when we ask a question, we understand the answer that's coming out. Especially using animals, this is not issue-free. It has ethical implications. Some people are very concerned about using animals in research. I will say that in the United Kingdom, where I have my laboratory, we have very high standards of animal care, and we have vets that come and see our animals, and we require licenses, and we have to justify all our work. So the primary model system that I use is the zebrafish, and this is a couple of centimeters long fish. It's from India, lives in the Ganges, and you've probably seen it in pet stores. They're very nice. They have this beautiful stripe pattern. And they were identified as a really excellent model system, or they were developed into a model system by George Streisinger in Oregon, because he wanted to study early development. He wanted to study an animal that had the vertebrate body plan like we do, but didn't want to use a mammal. He wanted something that we could use lots of different animals. And one of the advantages of the fish, which is a very strong advantage, is that the eggs are fertilized outside of the mother. So we don't have to kill a mother to look at early developmental stages in the mouse, for example, but in fact the female deposits eggs and the male then fertilizes those eggs, and we can collect those eggs from the tank. So it's we don't have to intervene too much in the animal, so it's excellent that way. This is an example, this is a schematic of what a zebrafish life cycle looks like. So we're able to see them at the single cell stage. So that's pretty incredible that that we can look at vertebrate life from the single cell stage and see that first cell division, see how this animal develops through thousands of cell divisions and undergo these shaping of the body plan. And within 24 hours, that fish has brain, eyes, and has melanocytes, these important cells that we know are so important for melanoma. And then in 3 months, it's an adult, and that adult can breed and generate hundreds, if not thousands, of these embryos. This is a little movie. This is capturing these first early stages of vertebrate development. This is the embryo here, and this is the yolk. You can see these cells dividing and dividing and dividing, and you'll get this little ball of cells on top, and then you'll see this dramatic shape change in the embryo as it starts to make tissues. Here we're going to see the development of eyes and a brain, here the tail, and you'll see the beginning of a vertebrate axis here. And that's all within 18 hours and just looking under a light microscope. So it's very powerful for understanding normal biology. And in the last 10 years, it's become very powerful for understanding disease biology. This is what our Zebrafish Lab looks like. We have hundreds of tanks of fish, and we collect them with nets, just like you would if you had a fish tank at home, and we collect their eggs mostly in tea strainers. So it's pretty low tech at that level. We keep different families of fish in each one of these tanks, and that allows us to keep track of our genetic systems. So how can zebrafish help us discover new melanoma therapies? Well, there's two important things I would like to emphasize today that I use in my research. And the first is, again, this concept that it's the melanocyte that's giving rise to melanoma. And actually what we've learned in the last 10 years is that melanomas really do know that they're from melanocytes, and they tend to reprogram these developmental programs inappropriately. And so development is one thing we can study really well in zebrafish. Here's a zebrafish embryo that's about 48 hours old. Here are the melanocytes. You can see them all nicely aligned and over the yolk sac. And if we use a transgenic marker, what we can see are in these green cells are these early stages of developing melanocytes. And these express a gene called MITF, the master melanocyte transcription factor, and this is what's required to make a cell a melanocyte. The other thing that's important that I'd like to emphasize is that when you have a melanoma, human or model one, not all the cells are the same, so that's called heterogeneity. And what we really are interested in is understanding how these developmental lineages become rewired inappropriately and end up with these large melanomas in the fish. So I'm going to tell you two short stories today that is about fishing for new therapies. And the first is to show you a model that we've created in the lab, and it's pointed us to a new cell population that we think we want to try to target. So my work in melanoma started when I was a research fellow in Len Zon's lab at Harvard Medical School. And at that time, the BRAF mutation had just been identified as being mutated in half of melanomas, and also moles, also in benign moles. And so what we did is we took that human gene and we put it into zebrafish. And what we found is that gene could drive moles and melanoma. And this was the first indication in an animal that the BRAF mutation could actually promote melanoma. So that was a very important and exciting discovery. Since then, of course, there's been fantastic work and drugs to target BRAF, and our understanding is much deeper and richer about the genomes of melanoma. This is a paper from 5 years ago, but it's still one of the landmark papers, and it has been updated. This is from the Cancer Genome Atlas Network. What they showed is it's not just BRAF that's important, there's also RAS mutations, NF1 mutations, and a group of patients for which we're not quite sure what the main driver mutations are. But what they also showed is that independently of these mutations, melanomas take on states, and these are called transcriptional states, and this is at the RNA level. And these can have what they call immune-like signatures, or they can have a keratinocyte signature, or they also have this signature which is called MITF low. And remember that MITF is that important transcription factor that tells you it's going to be a melanocyte. So we wanted to understand this MITF low group independently of these other mutations. And we knew we could, well, we thought we might be able to model this because we had a mutant in the lab, a genetic mutant, that had an MITF low activity. And so this fish is not able to make all the stripes it's supposed to. It doesn't have as much MITF. But it can make some melanocytes. But what's really important is that it's what we call a conditional allele. So we can turn it on and off. And in this case, just by changing the temperature of the water. And when we turn it off completely, we lose all the melanocytes. So this is a really powerful tool to look at this MITF low state, but also to ask, is it important for a melanoma to still know it came from a melanocyte? We also find if we cross this to a fish that has the BRAF mutation, we now get more melanomas more quickly, and they're more aggressive. Oh, sorry, we found we could get, we found if we had this mutation, we could get melanomas and in a BRAF independent fashion, and when we crossed it to a BRAF fish, we could get more melanomas that were more aggressive. So I mentioned that one of the advantages of this mutation is that we can turn it on and off. And so we then could ask the question, what if we turned off MITF once a melanoma was established, if we took away its identity? So here we have a fish that has melanoma. It has this low MITF activity. And I'll say these results we see both in BRAF melanomas and in non-BRAF models of melanoma. When we turn off MITF, we see these melanomas just disappear. So they rapidly regress. And the fish looks all normal, except it doesn't have any stripes because it doesn't have enough MITF to make its normal stripes. So this was very exciting. However, when we turn it back on MITF, the melanomas come back and in the same place. And so this, of course, makes us think of things like some of the responses to therapy. But it also made us think, maybe we really didn't cure this animal, and maybe there's what's called residual disease left. How could we find that? So what we did is we were able to label our melanoma cells and then take them through this protocol and then look by imaging, and what we can find is there's not many, but there are cells that remain at these regression sites. And we've been able to label them in such a way that we can even see that they're not all in the same cell state. So I think we've found an important population of cells that we need to target. So what are these cells? What we did is we took cells from a melanoma, this regression site, and recurrence, and we did this technique that you saw earlier, which is called single-cell sequencing. We looked at the transcriptomes of all of these cells. And what we found is that in general, melanomas that in the primary state look similar, cells in the residual disease look similar, recurrence looks similar, but we found this one state that was present throughout the progression of the melanoma. And to show you what that looks like, here in red, I've labeled the primary tumor. When we look at residual disease, we can see some are in these distinct states, but some are also shared in this common state. And again, with recurrent disease, we see a distinct state and a common state. So what that tells us is that there are multiple cell states, and some of them are present right at the beginning of that melanoma. So the key finding I want to emphasize is that melanomas are dependent on this master melanocyte transcription factor, MITF. They care about their identity for survival, so the bulk of the tumor went away, but some cells are independent of this, have developed independence, and this enables the melanoma to grow back. And the big picture concept I would like to communicate is that melanomas are comprised of subpopulations, and they have different survival potential, and so we're going to need to think about therapies to target these different cell states. So in the second part of my talk, I would like to tell you about how we're trying to target some of these cell states. And in particular, a cell state that we consider, this is a bit controversial in term, but a stem cell-like state. It's probably a bit naive to use that term, but that's how we generally try to think of it. And in fact, zebrafish are fantastic for drug screening, and this is really my great passion, is chemistry and biology using zebrafish. And this is because I told you that zebrafish have these highly conserved targets. So the genome is very similar between zebrafish and humans. So if we add a BRAF or MEK inhibitor to a zebrafish, it targets the same molecules in the fish. They have conserved drug metabolism, physiology, and pharmacology. And this has been a really powerful driver for new drugs. So this is an example of one of the screens that we've done. So what I did was to set up pairs of fish, male and female, capture their embryos, and put those embryos into plastic plates, literally sort of this size. And then in each of those wells in the plate was a different drug. And one of the families of drugs we found reduced the numbers of melanocytes. So here's a little zebrafish about 5 days old. Here are the melanocytes, these black cells here. And we can see quite clearly that there's fewer in this animal. And these drugs fell into one chemical class called the 5-nitrofurans. And these have been used clinically for over 40 years as antibacterial and parasitical prodrugs. So what prodrugs means is that they have to be bioactivated to become active, and their relative specificity comes by what's called a nitroreductase in the bacteria or the parasite. And this young child has Chagas disease, which is a trypanosome infection, and he's the type of patient who would receive these types of drugs. They have recently been found to have anti-cancer activity as well and are in phase 2 clinical trial for relapsed neuroblastoma and medulloblastoma, but why they work in cancer is really unknown. So we were very excited to get this class of molecules from our work, and we did a series of biochemical tests and identified a new target for the drug. And these targets are an enzyme class called aldehyde dehydrogenase. And what we know, but don't fully understand, is that in melanoma and many other cancers, there's a subpopulation of cells that express high levels of this enzyme. And those cells in the lab, when we put them into mice or other assays, seem to have more growth potential. We don't really understand too much more about it than that. But what we found is that these drugs can be bioactivated by this enzyme, leading to a toxic metabolite to killing those cells. So we thought this was an interesting opportunity to explore further. So here we use a mouse system where we've injected mice with melanoma cells and treated with this drug, and we can see that it doesn't allow, when we treat with the drug, the tumor doesn't grow as large as when we don't. But what's really important is that when we look for these ALDH high cells that we see in the melanomas, they're gone when we treat with the drug. So that tells us that we're getting to this target. The second piece of important data is I mentioned that these cells have these properties that we don't fully understand, but they seem to be better at initiating new tumors. And what we find is that if we, following this treatment that I just showed you, if we take those cells and inject them into new mice, but don't give them any further treatment, what we find is that the cells that had not seen the drug are able to grow new tumors, but the ones that had not seen, but the ones that had seen drug do not grow new tumors. So we haven't killed all the melanoma cells, but we've killed a subpopulation that's important for stimulating a new tumor. So we think that's kind of exciting. And we think this could have relevance to patients. We had the good fortune to collaborate with a clinician who could provide us with samples of melanoma before BRAF inhibitor treatment and after BRAF inhibitor treatment. And what we see in the pink is the level of ALDH enzyme. And we can see that this is enriched post-BRAF therapy. It wasn't enriched in all the patient samples that we looked at, but in about half. And so what that tells us is that for some patients, they seem to be upregulating this pathway following therapy, and maybe this would be a good population to target. And so the key findings here is that this class of antibiotics can target and kill this ALDH high cell population, and that for some patients, this might provide an opportunity to think about targeting this target. So the big picture concept I would like to convey is that not everything has to be a mutation to develop a drug against it. Sometimes it can be a property. In this case, it's a stem-like property that we're targeting. And that the other important concept is we have to do research on melanoma, but we also have to do a lot of research on the drugs. So we have to understand how drugs work. There's very few drugs, if any, that only do one thing in a human body. And so if we can understand the targets of these drugs through chemistry and through chemical biology, this widens their potential for use in treatments in other diseases. And so I hope that I've been able to convince you that model systems are really critical for bridging this gap between basic science and clinical science, and they really provide a very important function in this translational science window. And we think this is so important that actually we have a workshop on Friday exactly addressing the question of how can MRA researchers use our model systems to best address the most important clinical questions and clinical needs. And we do hope to have an output from that workshop in the form of a publication and perhaps even on the website, so we hope we can inform you about some of our discussions in that way. And with that, I would like to thank you so much for listening and thank my wonderful team, and also thank MRA for really supportive funding, both in terms of the funding, but also in the networks of people that they provide is excellent. So thank you very much.
We do have time for probably 2 quick questions. Does anyone? Yeah, Michael.
Thank you so much, Liz, for the presentation. It was great. You talked all about animal models, and I'm curious a little bit about organoids, artificial models, things that are being grown to serve as models, and wondering if you could just say a few words on that.
Yes, so these are other types of tissue. So basically, taking, for example, a human patient tissue and developing what are called cell lines or organoids, so being able to grow those in the clinic, in the lab. So these are also a really wonderful tool. First of all, they're human, so that really allows us to address human biology. And I think they're particular. We use cell lines a lot in our lab to really look at response to drug treatments. And indeed, one of the pictures I showed you was a cell line, and we could see that subpopulation that was high in that ALDH. No one model is going to be able to answer all our questions. Of course, a cell line is not a patient. You know, a fish is not a human, but it has more of a body structure than a cell line. So I think that kind of summarizes a little bit about why we really need to get our model systems people together and try to work collaboratively to answer some of the big questions that we have for patients.
I'd like to ask everyone to give Dr. Patton another round of applause.